next step is a fertilization. So fertilization, so how the fertilization happens? It is the fusion of the male and female gametes. It results in the formation of the zygote, which is 46 chromosomes. What we have seen, both gametes are haploid in nature. So ultimately, there has to be the chromosome number should come back to the 46, which will happen at the time of fertilization. So it will result in the formation of the zygote having 46 chromosomes. It initiates the embryonic development. What is the site and timing? These are the questions which are asked. Ampulla of the fallopian tube is the commonest site of fertilization. When does it happen? It happens within the 24 hours of the ovulation. The sperm's viability is also asked in the examination. The sperms are viable up to 5 days into the female genital tract. The oocyte is viable around 24 hours. Okay. So these viability periods are important from the examination point of view. Then what are the prerequisites for fertilization? First thing is there should be ovulation. Second, there has to be the sperm capacitation in the female genital tract, and there should be the intact zona pellucida, which will be penetrated by the sperm. Okay. So what are the steps of fertilization? Okay. So the first step is a sperm transport and capacitation. It happens in the uterus and the fallopian tube. There is a removal of the glycoprotein coat. Okay. So there is a glycoprotein coat around the spermatozoa that is removed, and there is an increase in the sperm motility and the membrane permeability, and this enables the acrosome reaction. Okay. So there is a acrosome reaction. As we have seen that there are certain vesicles which will contain the hydrolytic enzymes which are there in the head portion of the above the nucleus. There is a vesicle and they contain hydrolytic enzymes. Okay. So there is a next is a acrosome reaction. So this is triggered by the contact with the zona pellucida. Okay. So there is a ZP3 receptor. Once there is a contact with the zona pellucida, there is an acrosome reaction. There is a release of enzymes such as hyaluronidase and the acrosine. So which enzymes are important? Hyaluronidase and the acrosine. These are the enzymes which are present that allow the sperm penetration. Okay. So there is a penetration of the oocyte through the there is an outer covering called as corona radiata. Then this is the zona pellucida. So the corona radiata are nothing but the cells of the follicle. There are cells over here. So they form the corona radiata. Then there is a zona pellucida. And then after that there is a fusion with the oocyte plasma membrane. Okay. So inside this there is a plasma membrane, and the plasma membrane of the sperm will get fused with the plasma membrane of the ovum. Okay. So that is how it gets formed. After this, what happens in this space in the oocyte, there is a perivitaline space, and in that space there are cortical granules. Now what is the importance of this cortical granules? Here they are asking the questions. So there is a cortical reaction. They will block the polyspermy. Okay. So there is a release of the these cortical granules, and with that release of the cortical granules, this zona pellucida becomes very hard, and it will prevent the entry of the additional sperm. Okay. So what is the function of this cortical reaction? It makes this zona pellucida very hard. So once one sperm has entered into the oocyte, there will be no further penetration. Okay. So this cortical reaction is also asked in the examination. Okay. So then there is a completion of the meiosis 2. As we have said that the oogenesis, the meiosis 2 will get completed when there is a fertilization. So here becomes the completion of the meiosis 2. The secondary oocyte completes meiosis 2, and there is a formation of the second polar body and the formation of the ovum. Okay. So formation of the zygote, the male and the female pronuclei will fuse. This is called as syngamy. There is a restoration of the diploid chromosome number, that is 46, and this determines the genetic sex. Okay. So what are the key outcomes of the fertilization? So once the zygote is formed, after the fertilization, the zygote gets formed and it initiates the initial process of cleavage. Okay. Then there is a determination of the genetic sex. There is an activation of the embryonic genome and formation of the new individuals over a period of time. That is what is the outcome of the fertilization. So what are they asking in the examination? Zona pellucida, important in the prevention of the polyspermy. Failure of the tubal transport may lead to ectopic pregnancy. Fertilization outside the ampulla is rare, and IVF bypasses these tubal factors. Okay. So these are the things which can be asked in the examination. Okay, so once the zygote is formed, okay, we know that there is a male and female pronucleus. They will unite, and two cells will unite, will form the zygote. There is a fusion of the genetic material that creates a diploid zygote. There is a first mitotic division which begins. Okay, so early cell division, that is, they are called as cleavage. Okay, early cell divisions are called as cleavage. These are rapid mitotic division, and there is a formation of the blastomeres. Individual cell is called as blastomere. So this is the zygote, and there is a first cleavage. Okay, and these individual cells are called as blastomeres. Okay, these two cell stage, then there is a 4 cell stage, then there will be 16 cell stage. Okay, that is how there will be further division. Okay, so the morula stage is around 16 to 32 cell stage. So this morula stage is around 16 to 32 cell stage. There is a, after this, the from morula it will become the blastocyst. This is the hollow ball of cell which gets formed. It differentiates further into inner cell mass and the trophoblast. So this morula will develop a cavity, and after that it will become, it will have the cells will go at one particular pole and it will form the inner cell mass, while the cells which are covering these particular hollow cells of balls, that will form the cytotrophoblast, from which you are going to get the placenta, while the inner cell mass will give rise to embryo proper. Okay, so once this blastocyst is formed, the next step begins the implantation. Okay, so there is a preparation for the implantation. The blastocyst will reach to the uterus. Okay, then there is a zona hatching which occurs for the implantation. Okay, so from zygote, from day 0 to 1, single cell is formed after the syngamy. You have a diploid cell having 46 chromosomes, which is surrounded by the zona pellucida. Genetic sex is already determined, and there is no increase in the overall size of the cell as such. Okay. But from here onwards, what is going to happen? The stages what we have seen. So there will be further division of the cells, and these blastomeres will be formed, and you will get morula, then you will get the blastocyst. The morula is typically 16 to 32 cell stage. Okay, so what is morula? These are all the questions which are asked in the examination. Cleavage usually gives rise to solid ball of cells. Morula having around not 8 to 16, few authors say 16 to 32 cells. Okay, zona pellucida still forms the outer cover. Morula undergoes compaction, and the outer peripheral cells are smaller with tight junction. So there are two types of cell. The outer cells, which are smaller with tight junctions, while the inner cell mass consists of slightly large rounded cells with gap junction. Okay, so the inner cells are slightly larger, they are rounded, and they have the gap junction, while the outer cells are smaller and they have the tight junctions. The blastocyst formation. A blastocyst is comprised of 3 distinct sections: the inner cell mass that will develop into the embryo, a surrounding outer layer called the trophoblast that will develop into the placenta, and a fluid-filled cavity called the blastocyst. And at this stage, there will be implantation. Okay, if you see under the microscope, you will this zygote, the morula, looks like this. Okay, so they may give these particular figures also, and may ask you the stage of the embryo, stage of the development. So you look what exactly you see. If you see the blastocyst cavity, okay, if you see this one at inner cell mass and then peripheral cells, then you say that this is the blastocyst. Okay, you should be able to identify the morula stage and differentiate it from the blastocyst. Okay, so once this blastocyst is formed, the next step is an implantation. The final stage of early embryo development is the implantation of the blastocyst into the endometrial lining of the uterus. The blastocyst will breach the jelly coat that was covering it and preventing its attachment to the endometrium. So this blastocyst has got a jelly coat, and this jelly coat will prevent its attachment to the endometrium. So that should be breached first. Okay, so digestive enzymes are released which will degrade the endometrial lining, while autocrine hormones are released from the blastocyst that will trigger its implantation into the uterine wall. So it should happen at both places. One is the uterine lining should get disturbed, and second, this jelly coat should also need to be breached. So these are the two important stages which are happening. Only once the blastocyst is embedded within the uterine wall, it can the next stage of embryogenesis can happen. The growing embryo will gain oxygen and nutrients from the endometrial tissue fluid, ensuring its continued development. Okay, so now this embryo which is getting implanted will get its oxygen and nutrients from the endometrial tissue fluid. Okay, the entire process from fertilization to the implantation roughly takes around 6 to 8 days. Again, this they can ask you this question, the timing. This from fertilization to the implantation roughly takes around 6 to 8 days. Fertilization happens with generally within 24 hours, but till the implantation, generally it is around 6 to. Dr. Sansi?
Yes, sir.
Yeah.
Yes. Which one of the following statement about morula in humans is correct? It has far less cytoplasm as well as less DNA than in an uncleaved zygote. It has far less cytoplasm as well as less DNA than in an uncleaved zygote. It has more or less equal quantity of cytoplasm and DNA as in uncleaved zygote. It has more cytoplasm and more DNA than an uncleaved zygote. It has almost equal quantity of cytoplasm as an uncleaved zygote, but much more DNA.
Answer
Answer
is
is
also
D,
given.
sir.
Yeah, answer is also given
Yes.
here. It has got a equal quantity of the cytoplasm as an uncleaved zygote. Okay, but much more is a DNA. Okay, that is what is the difference. So very minute reading is required for the solving these SBAs. Okay, we feel that the
Yes,
information
sir.
is relatively easy, but these minor things you need to read very carefully. Okay, so cleavage refers to the rapid cell divisions that turn a single cell called zygote into multicellular structure. The daughter cells formed by the zygote cleavage are referred to as blastomeres. This all we have seen, okay, and we understand it easily. But when it comes to the question, we find it difficult, okay, because we are not reading in between the lines. The first zygote cleavage creates two cells that then split to form four cells and so on. A morula is known as a 16-cell embryonic stage. The rapid division of the cell during the zygote cleavage is not followed by the increase in the cell mass. Okay. Then this would be due to the short interphase resulting in decrease in the size of blastomeres as the total cytoplasmic mass remains close to that of the zygote with increasing numbers. Okay, so only cells will remain increase, but the overall mass will remain constant. Therefore, the morula has got about the same cytoplasmic weight as that of the zygote. Each cleavage division is accompanied by DNA replication with an equal allocation of the doubled DNA to daughter cells. Each cell division thus doubles the morula's DNA content. Hence, the answer is D. Okay. Now, next question. Oh, yeah, yeah.
Yes, yes, sir.
Yes.
The correct sequence of embryonic development is blastula, morula, zygote, gastrula, embryo. Zygote, blastula, morula. No, this is wrong, sir. Zygote, then morula, then blastula, then gastrulation, then embryo. Sir, option C.
Correct. Okay. What happens generally, two or three you can, but when they are combining three or four options together, no, we may get jumbled. Which stage happens? So correct sequence knowledge of that is how actually they are testing in the examination. Okay. So this answer is also given zygote, morula, blastula, gastrula, then there is an embryo. Okay. So all cleavage divisions are mitotic and resultant daughter cells are blastomeres. During cleavage, there is no growth in the resulting blastomeres and the total size and volume of the embryo remains same because the cleavage divisions interfaces without growth phase. That is, I think, the explanation for the previous question only. Okay.